So today I would like to discuss about enzymes. Enzymes are basically biological catalysts. They are used for enhancing the rate of chemical reactions by lowering the activation energy. So enzymes are mainly proteins. So proteins are compounds. We all know proteins are compounds formed by combining amino acids. They are macromolecules, and they will be having specific shape and all. And however, some of the enzymes are RNA molecules. They are termed as ribozymes, and they are found in the early stages of evolution and all. So broadly, enzymes can be classified as proteins. So in addition to this protein chain or RNA chain, there can be some other metal ions or something present in that particular enzyme, or it can be an organic molecule. So all these things will be termed as cofactors. So there will be a protein structure as well as a cofactor, which will be together giving the particular macromolecule which is having that specific catalytic nature. So two important features of an enzyme is the catalytic efficiency and specificity. The specificity of the enzyme arises due to the particular shape of the protein. So when we say that particular enzyme is specific to a particular reaction or particular analyte or particular type of process, we say it's not the full region of the enzyme that is showing, that is reason for the specificity. Instead, a small region in this enzyme, which is termed as the active site, which actually gives the specificity for that enzyme. Or this small region is where the particular reaction or that of biological importance is taking place. And this small region of the enzyme, if we are considering the enzyme carbonic anhydrase, we can see it is having about 259 amino acids. Only 4 or 5, or exactly 4 or 5 amino acids would be present in the active site. So this particular structures of the amino acids and particular orientation of amino acids will be giving the active site a specific geometry, and we can consider this active site and substrate combination as a complementary kind of bonding. That is, if you are trying to put on your left shoe on your right leg, it won't be matching. So similarly, only a specific substrate would be suitably fit onto this particular active site and able to undergo chemical reaction. And this binding of the substrate and the enzyme would be occurring due to by virtue of some kind of non-bonding interactions, such as hydrogen bonding, dipole interaction, pi star bonding, etc., pi bonding, etc. So some kind of supramolecular or non-covalent interactions will be enabling the enzyme to attach with the substrate and to catalyze a particular reaction. So the specificity of this enzyme is due to the complementary nature of the substrate and the active site. Second thing is the catalytic efficiency. Catalytic efficiency of the enzyme can be seen is the basic factor of the enzyme. That is, the enzyme would not be undergoing any chemical change during this process, and uncatalyzed reaction will be having a very much lower rate compared to the catalyzed reaction. Here you can see the rate of carboxypeptidase, carboxyl anhydrase, and ketosteroid isomerase enzymes, and the rate of catalyzed as well as uncatalyzed reactions. So how does this enzyme work? That is a very important question. Enzyme action can be simply looked into like this. This particular amount can be considered as a reactant, and reactant has to move all the way to form the product, and the movement of this man along the mountain to reach the other side can be considered as the particular process occurring during a reaction. So if he has to move all this way, he have to supply large amount of energy and move all this way like this. But suppose we have another path, we have another way, like a tunnel or something through which he can easily get to the other side. It will be very much energy efficient, and it will be requiring less effort and less energy. And this is particularly what enzyme does. Enzyme provides an alternative pathway, and a particular pathway where energy requirement is lesser than the actual pathway. So when you are talking about a reaction pathway or a chemical reaction, there are two important thermodynamic features. The first one is the free energy of the reaction, and the free energy difference between the reactants. So the product and the reactants will be having difference in free energy, difference in enthalpy, etc. And this difference in free energy can be used to calculate whether a particular reaction that we are considering or trying to do is feasible or not. So if the difference in free energy between the products and the reactant is negative, then we say that reaction can be done, or this reaction is spontaneous and can be easily done. But some of the reactions, even though they have negative delta G value, they won't be easily occurring, and this is due to some other principle, that is the activation energy of a reaction. So a simple example is, if you consider the oxidation of sucrose to carbon dioxide and water, this reaction occurs in our body in every milliseconds, that is during the respiration and all, this particular process is occurring. But we cannot see, or we cannot actually observe the conversion of sucrose to carbon dioxide and water in presence of oxygen. When you're keeping the sugar in your plate or in your kitchen, it won't be undergoing this kind of change. So the reason for this particular this conversion is actually having a delta G value negative, but it is not spontaneously occurring. So the reason behind this is the particular reaction requires a high activation energy. So this activation energy barrier is overcome in our body due to the presence of enzyme. However, in our daily life, we won't be observing a sugar that is kept in a spoon or plate, not undergoing any chemical oxidation to carbon dioxide and water in the absence of enzyme. So what enzyme does is it just lowers the activation energy. If you're considering a reaction, you can see the reaction would be proceeding towards an equilibrium, and enzyme does not change the position of the equilibrium. Position of the equilibrium will be giving you the equilibrium constant, and this equilibrium constant is not affected by the presence of enzyme. Instead, enzyme will be changing the rate at which the particular reaction reaches the equilibrium condition. So the reaction rate is altered. Reaction rate is done by lowering the activation energy of that particular reaction, and this lowering in activation energy is achieved by providing an alternative pathway, and this alternative pathway includes an enzyme substrate complex as well as enzyme product complex. So the mechanism of enzyme action can be briefly said like this. The enzyme first binds to the substrate to form an active enzyme substrate complex, and it will be released to form the product. So for any enzyme catalyzed reaction, the important step is the formation of enzyme substrate complex, and this complex will be having the interaction, having various non-covalent interaction to bind the enzyme and substrate together. So here, when enzyme combines with substrate to form enzyme substrate complex, it is a reversible process, and enzyme substrate complex decomposing to enzyme and product is a reversible process which is having a rate constant K2. So the formation of enzyme substrate complex is important aspect in mechanism of enzyme action, and this formation of enzyme substrate complex was verified and confirmed by various techniques such as kinetic studies, X-ray studies, and spectroscopic studies. And from the kinetic studies, what the scientists did was they followed the rate of the reaction, initial rate of the reaction, with the concentration of substrate. As the concentration of substrate was increased, they found that there's a rapid increase in the rate of reaction up to a certain value, and then it slowly the change in the increase in the rate of reaction was very much smaller, and then after a certain concentration of the substrate, the rate of reaction remained constant. And this kind of plot, or rectangular parabolic kind of plot, was indication of the formation of some species that is limiting the rate of reaction, and this is due to the formation of enzyme substrate complex. However, concentration of substrate you increase, there is a specific species that is formed. That species concentration is limited by the amount of enzyme that is present. So if you have 10 enzyme, only even if you provide 100 substrates, only 10 will be combining at a particular time to form the enzyme substrate complex. So that particular enzyme substrate complex formation limits the particular reaction. And the second evidence was from X-ray spectrophotometry, and using X-ray crystallography, it was possible to isolate some of the enzyme substrate complexes at very low temperatures and study the corresponding structures and all. And using spectrophotometry or spectrofluorimetry, the scientists were actually able to identify the formation of various enzyme substrate complexes. Here you can see the formation of enzyme substrate complex with a serine and enzyme is given here. Tryptophan synthase catalyzes the synthesis of L-tryptophan from L-serine. So by fluorescence studies, it was found that in presence of serine and enzyme, the fluorescence intensity was largely increased. However, in the enzyme alone or the serine and product alone could not give that increased fluorescence intensity. This indicates that there is some kind of binding between this serine and enzyme, which increases the fluorescence emission of that particular tryptophan residue. The kinetics of enzyme action was studied by various chemists and various scientists, and two among them was Leonard Michaelis and Maud Menton, and these two actually proposed the concept that enzyme would be first binding with the substrate to form enzyme substrate complex. So then they derived a kinetic model for enzyme substrate complex formation and enzyme action, and this particular model is termed as Michaelis-Menten model, and the equation obtained in the corresponding study was Michaelis-Menten equation, which is a very simple model for enzyme and useful for many kind of enzyme. So what they did was they studied the initial rate of the enzyme catalyzed reaction at various concentration of substrate. So they observed that as the substrate concentration was increased by keeping the enzyme concentration constant, the rate would be increasing first and reaching, and first it will be rapidly increasing, then it will be slowly increasing and reaching a maximum value. And then the graph of initial velocity versus concentration of substrate, which actually gave a rectangular parabola, and it is termed as Sternwarmer plot. It is termed as Michaelis-Menten plot, and from this particular graph we obtained the equation for Michaelis-Menten equation, that is V0 is equal to Vmax in the substrate concentration divided by Km plus substrate concentration, where V0 is the initial rate and Vmax is the maximum rate and Km is the Michaelis-Menten constant. So when the S value or the concentration of substrate is very high, V0 will be equal to Vmax, and when substrate concentration is very low, V0 will be equal to Vmax into S divided by Km, and when V0 is equal to Vmax by 2, that is half of the maximum velocity, in this Km will be equal to substrate concentration, or in other words, the Michaelis-Menten constant is the substrate concentration at which the initial velocity will be equivalent to the half of the maximum velocity. So the Michaelis-Menten equation give significant information about the stability of enzyme substrate complex, and it will be helpful for finding the rate constant of each of the reaction steps, and it can be useful for calculating the turnover number, catalytic efficiency, fraction of active site filled, etc.